Hi guys, today we're going to use Skittles to figure out how to do chi-squared calculations. We're going to do a candy lab today, so I just wanted to go over that with you. Okay, so what is a chi-squared test? It's this math thing that we do. It's a test to see if the relationship between two variables is statistically significant or if it's just due to chance. The product is known as a p-value, and we're going to talk about p-value a little bit later. Okay, so the first thing that we have to do in our lab here is we have to come up with a hypothesis. We always start out with what's known as the null hypothesis. There will be an equal amount of each color of Skittles in the bag. So we start off with a bag of Skittles. We're going to say that the dispersion is equal. My personal hypothesis is that there will be fewer of the citrus flavors, honestly, because they're gross. So I'm going to say that I'm going to try to reject the null hypothesis because I don't think it's wrong. I don't think there are equal amounts in the Skittles bag. Okay, so what we have to do is we're going to have to start counting. So you have your bag of Skittles, dump them all out, sort them into colors, and count how many of each color you have. You can push pause if you need to. So I'm at home and I actually did this. So I sorted them all out, and just like the data table that you have in your lab paper, I have my percentage estimate. Well, I have 5 different colors, so it has to add up to 100% down here. So 5 into 100 equally is 20. So that's where I came up with my 20%. So if they are distributed evenly, 20% of all of my Skittles should be red, 20% should be purple, 20% should be yellow, 20% should be green, and 20% should be orange. When I counted them, I did not get that. What I got instead was I got 38 red, 23 purple, 18 yellow, 25 green, and 40 orange. And when I counted them up, all of them together, I got 144 Skittles. The number expected that's over here is, if I said I was going to have 20% of my Skittles were going to be red, I just did 20% of 144, and that's where I got 29. So if all of them were equally distributed within the bag, I should have 29 of each color. That would be my number expected. And when I came down to the bottom, I had 145. That's just because I had a decimal point and I can't have like 0.8 of a Skittle. So I had to have a full Skittle, so I made it 145. Okay, now that we have that done, we need to do the math so that we can find our chi-squared and we can figure out if our distribution is due to chance or if our numbers really aren't equal inside of the bag. So I have my expected numbers over here, and all of my expected numbers are the same because remember, I said that our null hypothesis is that they were equally distributed. So I have 29 of each color. Now, what I actually observed, these are the ones that I counted. That's where I got my 38 and my 23 and my 18 and my 25 and my 40. Now I have to fill in this part over here. So I'm going to go to the next slide and I'm going to show you how to do that on the red one. So we're going to start with the red Skittles first. This little equation that you see up here, this is the O is observed minus the expected, and then we square that. And then we divide it by the number of expected. Now the nice thing about the fact that we square this is that if we end up with a negative number, it's never going to matter because it's the difference between the observed and the expected. So our observed number is 38. Our expected number is 29. So I took my 38 minus my 29. That gave me 9. I squared that. That gave me 81. And then when I took my 81 divided by 29, my expected, that gave me 2.79. So that's the number that I'm going to write in the next column over in your data table. Right here. That's my 2.79. I did the exact same thing for my purple, for my yellow, for my green, and for my orange, and that's what these numbers right here are. Okay? Now finally at the end, I'm going to need to find the sum of these. So if you look at your chi-squared formula, you'll have that weird little S-looking thing in front. That's the sigma. That means the sum of. So my chi-squared is the sum of all of these guys, which gives me a chi-squared of 11.54. Now that I have that, I have to find my p-value. And there really aren't any calculations I have to do with that. I just get to use a table, which is really easy. So here's the chi-square table. And if I look over here, see this degrees of freedom? We had 5 different colors of Skittles. So what you do is you take your number of categories that you have and you subtract 1, and that's your degrees of freedom. So I'm going to have 4 degrees of freedom because I had 5 colors of Skittles. Now I go over here and I am looking for what is closest to my number, which is this one right here. So my 11.143 is closest to what I came up with on my chi-squared table. And if I go up here, see how I have 0.025. It says poor fit. Don't let that mess you up. Poor fit is a good thing. Poor fit means we get to reject the null hypothesis. And this right here, this 0.05, this is kind of the line. If you're less than 0.05, then that's a good thing. If you were to the other side over here, then this means that you have a good fit to your null hypothesis, and so all of our Skittles would be equally distributed. So here's how we interpret the results that we get. If you have a p-value that's less than 0.05, then you can reject your null hypothesis, and your alternative hypothesis is likely correct. Not necessarily correct. For instance, the null hypothesis was that everything is distributed evenly throughout the bag, the same number throughout the bag, the same percentage throughout the bag. That part we have decided, well, we're going to decide, is not going to be correct. That's the null hypothesis. It's not correct. However, my alternative hypothesis, that there are fewer of the green and the orange and the yellow because I think they're gross, that's not necessarily correct. I'm going to have to do some more research on that one. If you get anything over 0.05, it is a poor fit, and you have to accept the null hypothesis. So you would accept that everything is distributed evenly throughout the bag, even numbers of colors. So a good fit, remember on that chart, if it was greater than 0.05, that was a good fit. So a good fit means that the null hypothesis is correct and there's no relationship between your variables. It's just due to chance. Our p-value is 0.025. All right, so what does that mean for us? Yeah. Okay, so we can reject our null hypothesis because our p-value is less than 0.05. Okay, my p-value is statistically significant. And the reason why it is because I was less than 0.05. I can reject my null hypothesis. That means that there are not equal numbers of colors. That means that there's a chance that my alternative hypothesis could be correct and that I need to do some more research on that. But it tells me that there is some kind of relationship. It's just up to me to figure out what it is. So I would need to design an experiment to investigate further. So this is all there is to chi-squared. It's not very hard. All it is subtracting and squaring and dividing, and then you add it all up. Look on a chart, and then you're done. All of these things, the equation, the chart, all of that stuff is going to be on your equations handout for the AP exam, so you never have to memorize it. You just have to know how to use it. That is all.